Hey, welcome back for Chapter 3. This is the basic intro to chemistry, and there's a common misconception that people think of science as separate disciplines, and in a way it is, and that's a good way to organize and concentrate your focus in specific areas, but when you boil it all down, everything is actually physics. Chemistry is applied physics, biology is applied chemistry, and so on and so forth. So you kind of have to understand a little bit of everything in order to understand anything. So we're going to go through basics of chemistry here, and don't worry, this and the bit on photosynthesis and respiration is all the chemistry you'll have to know for the course. So, matter is anything that takes up space and has mass. And yes, there are more than three states of matter. There's things like plasma. I'm sure most of you have heard of plasma at one point. That's the fourth state. But there's also weird things like condensates and just. But for our purposes, you just have to know the three: solid, liquid, and gas. And on a daily basis, that's about all you're going to interact with. All matter, living or non-living, is made up of elements, and that's a substance that cannot be broken down into simpler substances by ordinary chemical means. Review the periodic table of elements if you're curious about examples. But for our purposes, the main elements we're worried about are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. These make up the majority of organisms by weight. So the atomic structure. The smallest part of an element that displays the properties of the element is an atom, which is made up of three subatomic particles. You have the proton, the neutron, and the electron. At one point in time, we believed that the atom was the smallest thing that there was, that nothing could get ever any smaller than that, which is why it was named atom in the first place, from the Greek atomos, meaning indivisible. But then we discovered subatomic particles. So the proton is positively charged, located in the nucleus. Neutron, as the name implies, is neutrally charged and located in the nucleus. And the electron is negatively charged and exists in a probability field outside of the nucleus. And you'll constantly see this kind of model here where it's like planets orbiting. That is woefully inaccurate, but it is useful for getting the concept across as to kind of where it's supposed to be. For all intents and purposes, an electron has no mass, and a proton and a neutron are both one atomic mass unit. Though technically speaking, a neutron is slightly heavier than a proton, and the difference in their mass is the mass of an electron. But that is beyond anything you need to worry about for this course. If you look at a periodic table, you'll see the atomic symbol, so C for carbon, the mass number, which is the sum of the protons and neutrons, and the atomic number, which is the number of protons. You can have variance here, and we'll talk about that in a moment with isotopes, but the atomic number always tells you what element it is. And then you can use that in conjunction with the mass number to figure out which isotope you're talking about. The periodic table has certain trends that reoccur periodically, hence periodic table. The vertical columns are groups, and the horizontal rows are periods. The noble gases on the end here are essentially inert because their outer shell has 8 electrons in it and has completed the octet rule. And the main thing you need to know about a period is that all elements have the same number of atomic orbitals within that period. And another nice thing that may or may not be a question is that row 7 here can only interact with one atom at a time, and that's because they have 7 electrons in their outer orbital, so they can only form one bond at a given time. For different isotopes, you have, so this carbon-12 is like the most abundant. You see it all the time. Nothing particularly strange about it. Carbon-13 has one additional neutron in it. in the nucleus, and that just makes it a little heavier. And carbon-14, on the other hand, has 2 additional neutrons and is unstable because of that, which makes it radioactive. So when people talk about radiocarbon dating, what they're looking at is the relative percentage of carbon-14 in something relative to what it was believed to have been when it was buried or whatever. So in theory, the older something is, the less carbon-14, because it is radioactive and will decay into carbon-13 or carbon-12. We can use low levels of radiation for like thyroid scans to look, because the cancerous tissue is more metabolically active and will take up the irradiated iodine much more so than the rest of it, the rest of the thyroid. You can use radiation to sterilize things. You can kill bacteria and fungus with it, or you can use what's called a gamma knife to irradiate a very specific area with high levels of radiation to just straight up kill cancer cells in that particular area. A little bit risky, but very useful under certain circumstances. So in addition to the nucleus, you have electrons out here buzzing around it. If you're talking about a neutrally charged atom, the number of electrons will equal the number of protons. And they move in these orbitals outside it. And again, electrons are not really a particle. They behave more like a wave most of the time, but there's this probability field where it's most likely to be at any given point in time. So we kind of treat them like particles, but that's a little beyond the scope of this course. Few things you need to know. The first shell here always contains 2 electrons, and then every other one will hold up to 8. So carbon here, as you can see in its outermost shell, has 4 electrons, meaning it needs 4 more, or needs to give up 4, either way, for its outermost shell to be completed. And that means carbon is perfectly willing to form 4 bonds at any given point in time. Octet rule means that the outermost shell wants to have 8 in it. Some things only have this first, or some elements only have this first shell, in which case they want to have 2. Or if you're a hydrogen and you've only got 1 electron here, you're perfectly willing to give up that 1 electron and satisfy it that way. The type of bond that you will form depends on the electronegativity of your elements, but we'll get into that in just a moment. If you have 2 or more of the same element bond together, then we're calling that a molecule, which I know we'll also talk later, and we'll say like a molecule of glucose. So just for the purposes of this first exam, use this definition of molecule, and then we'll worry about the other later. And then if you have 2 or more elements bonded together, that's a compound. And when a chemical reaction occurs, energy is either given off or absorbed as you're making and breaking bonds. And you have multiple types of bonds: covalent, ionic, hydrogen. So ionic bonds occur when you have very different electronegativities between elements. So electronegativity is a measure of how strongly that element wants to steal an electron. Now chlorine here has 1 missing spot in its outer shell. That makes it extremely electronegative. Sodium here has 8 missing spots. So it's actually a lot easier for it to just give up that 1 electron and then have this other completed shell right here. So it is not, it's one of the least electronegative elements. So what happens then is the chlorine will steal this electron, which will result in the chlorine having a negative charge and the sodium having a positive charge, which means positive attracted to negative, so you have an ionic bond, and so they are just kind of like stuck to each other based on that charge differential. This forms sodium chloride, which is table salt. If somebody ever says salt, you need to clarify which one, because there are hundreds of types of salt, and most of them are toxic. But sodium chloride, table salt, you need that to live. Which is kind of interesting if you think about it, because chlorine gas will kill you if you breathe it in. And sodium explodes in contact with water, and we're about 70% water. So if you just look at the ingredients of table salt, you would think that would be incredibly poisonous. But you put them together and you get table salt, and you need that to live. So go figure on that one. Covalent bonding, when you have two things that are roughly equal in electronegativity, they will share the electron between them. And so as they're sharing, like hydrogen here, they both want two, and so this one brings one to the party, this one brings one, and they kind of bounce between the two, which is why when you find hydrogen, you never find monoatomic hydrogen. It's always H2 when it's just out and about on its own, because it's much happier like this. Same thing with oxygen. So O2 is how you find oxygen out in the environment. And it's just sharing these electrons back and forth. Now the thing about it is, electronegativity still comes into play here. So the attraction, which is the attraction of an atom for electrons in a covalent bond. So if it's a nonpolar covalent bond, there's an equal sharing, or mostly equal sharing between the two, like so. If it's polar, then there is an unequal sharing. And that will result in, like in water here, you have partial positive charges on the hydrogen and partial negative charges on the oxygen. Because a greater percentage of the time, the oxygen, being more electronegative than the hydrogen, is hogging the water. And then also, is hogging the electrons. Which leads to this, bent configuration. But it allows for hydrogen bonding. So because you have this partial positive and partial negative, you can have hydrogen on one water molecule kind of interacting with and being drawn towards the oxygen on another, and it makes water very cohesive. It sticks together quite strongly. And again, this is one of those things, if you just look at it, hydrogen gas plus oxygen, very good for fires. But you combine them into water and you'll put out a fire. So chemistry is funny that way. The end product is not necessarily what you would expect based on looking at the reactants or the properties thereof. Chemistry of water, we're between 70 and 90% water. Well, most organisms. Water is a polar molecule and can form hydrogen bonds because of that, like we just talked about, which makes water highly adhesive and cohesive. It sticks together because of the hydrogen bonding, and the positive and negative poles allow it to adhere to other surfaces, meaning adhesive. Excellent transport system, both within and without living organisms. High surface tension, thanks to all the hydrogen bonds, which allows some things to walk on the surface of it. It has a massive specific heat, which means, or heat capacity, which means it can soak up tons and tons of heat energy without changing states, which keeps our temperature, the temperature of the planet, very nice and stable from day and night, because during the day the water in the oceans is just soaking up massive amounts of heat, and then at night, when the sun's no longer shining on it, it releases it, so we don't have these huge temperature swings from day to night like you do on, say, Mars, for instance. It also has a very high heat of evaporation, which is why we sweat. You put water on the surface of your skin in a thin layer, it evaporates, and when it does, it dissipates a massive amount of heat from your system. Which is one of the advantages that we have of being mostly hairless. We actually are capable of cooling down more efficiently when we're doing something like running than when we're standing still. And so there are certain tribes in Africa who still practice a form of hunting where you essentially just run your prey to death, because over long distances, nothing is faster than a human, because we have this insane capacity to cool ourselves off and regenerate our stamina while on the move. Other animals with their fur are not very good at sweating, so they have to pant in order to cool down. Um, Dogs, for instance, only really can kind of sweat on their nose and maybe on the little pads of their feet, which is why you'll see a dog frequently just stop, lay down, and pant for a good while. He's doing that to cool down, not because he's out of breath. So over long distances, we can run anything to death, and it was a very useful adaptation for spreading across the planet. Water is also a solvent. Basically, everything dissolves in water to one degree or another, even things you wouldn't think about. Steel will dissolve a little bit in water. There's iron in water, these sorts of things. Some things obviously dissolve in it better than others. Oils typically that are very hydrophobic do not attract water. They're actually repelled by it, so they never actually get around to interacting with the water. So they're not going to really dissolve in water. But aside from that, basically everything does to one degree or another. Hydrophobic molecules attract water. And in case you haven't noticed, anything in bold as you're going through these PowerPoints, probably going to be a question on it. Water is kind of unique, and because of this hydrogen bonding, when it freezes, it forms this lattice work, which actually takes up more area and is less dense than the liquid variety, which is why ice floats. It also, when you evaporate it, it becomes a gas and takes up much more space, so it's kind of unique in that it's one of the few things that takes up more space and becomes less dense when it is heated or cooled than if it's in its liquid form. Which serves us well, because if ice was less dense than water and went down to the bottom of the lake, then every lake would freeze solid every winter and would kill just about everything living in it. Anything that releases hydrogen ions in water is an acid, and anything that releases hydroxide ions in water is a base. And the funny thing is, water can actually dissociate into both. So in the right context, water is both an acid and a base. And there are other definitions for acid and bases, but for our purposes, this is the one you need to know. Anything with a pH below 7 is acidic. Anything above 7 is alkali. 7 is neutral. Our pH is roughly 7.4-ish. And that's if you were to go a full pH out of that, 6.4, 8.4, that's no longer compatible with life. We go through a lot of trouble with buffers and such. The primary buffer we use is sodium bicarbonate. It's not that good of a buffer, but we have like tons of it in our blood, so it does the job. Which is why, like, if you ever hear someone telling you, like, drink alkali water or something to change the pH of your body, they're full of it. We have so many buffers and respiratory and metabolic systems to regulate the pH of our internal environment that you can drink alkali water all you want, and it's not going to change your pH in the slightest. You're just going to end up urinating or breathing more to compensate for it. And if you did actually manage to alter the pH of your blood, it would probably have dire health consequences. So bear that in mind. That's one of the examples I give when people ask me, well, where am I actually going to need to know this stuff? I was like, well, when some charlatan tries to sell you snake oil, you'll know that what they're trying to make you buy does not actually work according to the laws of physics or chemistry or biology. Save yourself some time, money, and health complications. Organic molecules are those that contain hydrogen and carbon. If you only have hydrogen carbon, you are a hydrocarbon. Again, scientists are very clever with the names. Organic molecules are or well, for organic molecules, the chemistry of carbon accounts for the formation of most organic molecules, which is why when you take organic chemistry, it's just tons and tons of this with different things on it. It's all about that carbon. You have different functional groups, and we'll try to zoom in here without breaking anything. Okay, so hydroxyl, carbonyl, carboxylic acid, amino groups, sulfhydride, and phosphate groups. Basically just kind of have a general idea of what these are. Hydroxy groups are formed in ethanols, alcohol, aldehydes, and ketones are both carbonyl groups. The way to keep this straight is aldehydes do not hide. They're just out here on the end, whereas the ketone is in the middle of something, just double bond oxygen here. Ketone bodies can be used as a alternative fuel source if you are in a starvation state or you're on a ketogenic diet. You'll kind of get that keto flu as you're switching over, because it takes about a week or so to get high enough levels of this in your system to properly fuel your nervous system. So you're going to feel like crap during that transition. Carboxylic acids, you just see them in things like acetic acid. Amino groups you're going to see in amines such as tryptophan. Tryptophan, by the way, does not make you drowsy, at least not when you're eating your Thanksgiving dinner. There's so little tryptophan in that turkey, and it's competing with so many other things to being digested and put into your system, that it's not responsible for making you drowsy. What is responsible for that is you've just consumed, 1,000+ calories, and your body is like, well, okay, let's go sleep this off and enter rest and digest. Sulfhydrides, again, very cleverly named. It has a sulfur and a hydrogen. Phosphate group, you're going to see so many phosphate groups when we start talking about cellular respiration and the kinase that phosphorylates the kinase, and on and on and on. Just understand that it's typically used as an energy carrier. And we'll try to return the zoom without breaking this. Okay, so organic molecules, 4 major groups. You've got proteins, carbohydrates, lipids, and nucleic acids. Monomers, simple organic molecules that exist individually. Polymers, large organic molecules formed by combining monomers. So, interesting thing to be aware of here. A carbohydrate is a polymer of a monosaccharide, meaning single sugar. So carbs are sugar. Proteins are polymers of amino acids, and nucleic acids like DNA or RNA are monomers of nucleotides. Organic molecules, you have you're typically forming these polymers through dehydration reactions. So you're removing a hydroxy and a hydrogen from the two ends of your monomers, forms water, and then creates a polymer. You break them by doing exactly the opposite, forming a hydrolysis reaction, to hydro water, lysis to break apart. So break apart with water, again, clever names. Carbohydrates, typically quick fuel, short-term energy. Now they play a structural role in woody plants and bacteria. The funny thing is the starch in your potato is exactly the same as the starch in a 2x4. It's like the subunits anyway, the monomers. It's just one's in an alpha configuration and one's in a beta. And you have the enzymes required to break down the potato, but not the 2x4, which is kind of cool, I guess. Simple sugars, monosaccharide, mono 1 saccharide sugar, and disaccharide contains 2 monosaccharides, di 2 sugar saccharide again, joined by a dehydration reaction such as maltose, sucrose, lactose, broken apart by hydrolysis. Technically speaking, a disaccharide is a polymer. It's just we have a specific name for when it's only 2. I say that because the next thing down here is polysaccharide, which is many glucose subunits. So I guess for technical purposes, 3 or more. But you have things, so that would be like starches, which is the form of glucose storage in plants, or glycogen, which is how we do it, and that's stored in the liver. Um, Funny thing, you can only store so much glycogen in your liver at a time, and if you have too high of a caloric intake that overwhelms this capacity, you'll start storing it as fat because that is a much denser storage medium for energy, which can lead to a fatty liver, which then leads to cirrhosis and cryptogenic hepatitis. And so a high sugar diet is just as bad for your liver as chronic alcoholism. Um, that and throw metabolic syndrome on top of that too, and it's a real mess. The average American consumes something like 10 times the daily recommended allowance of sugar. But if you look on your Coca-Cola, you won't see a percent for the sugar. Um, but I think at most you're supposed to consume something like between 30 and 50 grams of sugar, depending on your size. So, we'll just say 40 for the average. And a lot of soft drinks for a single can will have between 35 and 55 grams of sugar, depending on what you're imbibing. So, in one drink, you have consumed equal to or greater than your daily intake of sugar. And anything above and beyond that starts doing damage to your system. So bear that in mind when you go to the grocery store next time. Also, fruit juice is terrible for you. Avoid that at all costs. Fruit is good because it has fiber, so you're not dumping the sugar into your system, but the fruit juice itself has so much sugar in it, it's actually extremely detrimental to you. Same goes for small children. Glucose as the cell walls of plants, or is used in the cell walls of plants. I'm sorry, cellulose. I got sidetracked there. Cellulose is used in the walls of plants, accounts for strong walls, and has a different linkage than starch or glycogen, which prevents us from digesting it, but it is again the same subunit. This is all just sugar. And then chitin is found in the exoskeleton of crabs and is another structural polysaccharide. Lipids contain more energy per gram than any other biological molecule. Structurally, although they have a diverse structure and function, they have one common characteristic, and that is they do not dissolve in water. They're hydrophobic. You use there's fats and oils used for energy storage, phospholipids for your membranes, steroids including your sex hormones. Oils are typically of plant origin, liquid at room temperature. Fats are triglycerides, and are of animal origin, are typically solid at room temperature. And a triglyceride consists of a glycerol backbone and 3 fatty acids. The fatty acid is a hydrocarbon chained with an acidic group, typically a carboxylic acid at the end. Unsaturated fats have one or more double bonds, like so, and that creates this kink in it so they don't stack as tightly, meaning they have lower boiling points or melting points. And then saturated have no double bonds. The big deal with like trans fats, it has to do with the type of bond here, which way it bends or is kinked, and you'll get into more of that with organic chemistry later on if you take that. Maybe in Gen Chem, I don't really remember off the top of my head. But we don't have enzymes that function to attack trans fats and digest them for food as well, which is why despite everything coming out of a deep fryer tasting delicious, it's probably not good for you, because you have a greater rate of flipping these things over into the trans configuration instead of the typical cis configuration, meaning you can't digest them, and a lot of lipids are also pro-inflammatory, so yay. Steroids, typical examples include cholesterol, testosterone, and estrogen. They have this backbone of 4 fused carbon rings, and they're usually, messengers of some sort. They influence or direct the functions of tissues. Proteins are, well, they have a ton of functions. So they can support things, they can catalyze reactions, they can function as chemical messengers. Actin and myosin move your cells and contract your muscles. Some things are used as transport molecules in blood, like albumin. Albumin's basically your body's taxi driver. It just sends everything everywhere. And some of them, form antibodies to protect the cells and attack invaders, and others allow for channels to form to cross membranes. So, the proteins do all the things, basically. You can even break them down as energy. They're composed of amino acid monomers. So the amino group, you've got the NH2, like we talked about. Acidic groups, we've got our carboxylic acid right here. The R group varies, and an R group is basically just a carbon with whatever else attached to it. It could be anything, so you just abbreviate it as R, just to make writing it out easier. Peptides are chains of amino acids, or polypeptides are chains of amino acids, and they're joined together with a peptide bond. Proteins can have up to 4 levels of structural organization. And remember, form fits function. So the form that something takes defines what it does. And so the primary structure here is the linear sequence of amino acids. Thymine, lysine, whatever. You just form this chain, and some of them are hundreds or thousands of amino acids long. And secondary structure, you start to have these hydrophobic and polar and nonpolar interactions within it, and they'll form alpha helixes and beta sheets. Those then interact with each other to create this tertiary 3-dimensional configuration of the protein. And then multiple subunits of this will come together to form a quaternary structure, which is how all the subunits fit together. And typically this is the functional unit of the protein, though there are plenty that function just at this level here, and that's complete. But like hemoglobin, for instance, you need 4 different subunits functioning together in order to perform its function. Nucleic acids, you have 2 different nucleic acids. You have DNA and RNA, and the difference between them is which sugar you're using. So in DNA, it's deoxyribose, and in RNA, it's ribose. And both DNA or RNA and RNA are polymers of nucleotides and consist of a phosphate, a pentose sugar, and a nitrogen-containing base. And again, there's one of those little phosphate groups. You're going to see those things over and over again. So, structure DNA, you've got your sugar, so deoxyribose and ribose, your nitrogenous base, adenine, thymine, cytosine, and guanine, and you will see uracil only in RNA and thymine only in DNA. So keep that in mind. And then the structure of DNA is a linear form called a strand that produces this double helix, and the 2 sides of it, like the rung, are held together with these kind of rungs in the middle, which are bonded together with hydrogen bonds. And the rungs are formed by complementary base pairs, so A always bonds to T in DNA. In RNA, the T is replaced with a U, and then C always bonds with G, no matter if you're talking DNA or RNA. You have 3 types of RNA. You've got messenger RNA, transfer RNA, and ribosomal RNA. And this chart's pretty handy here. So you've got the different sugars, depending on which one you're talking about. Different bases. The only thing that's different is the thymine and the uracil. And RNA is single-stranded, except in viruses. And RNA does not have a helix. DNA does. So that's a bit of RNA right there. So then we have nucleic acids here. ATP, adenosine triphosphate, is a high energy molecule, undergoes hydrolysis and energy is released, meaning you cleave off one of these phosphate groups because it's adenosine triphosphate, 1, 2, 3 phosphates right here. You chop one of these suckers off and it releases a ton of energy, which is why this is the energy currency of the cell. And then that turns into ADP over here, and you have to undergo a variety of processes to rephosphorylate this to make it usable again, and we'll get into those in greater detail later during cellular respiration. And that is it for chapter 2. This is probably the chapter that most people have the greatest difficulty with, just because it's chemistry. If you wanted to take chemistry, you'd be in a chemistry course, but we have to do a little intro to chemistry so you'll understand some of the later concepts. So I recommend spending a little more time with this chapter, and if you have any questions, comments, concerns, feel free to send me an email, and I'll see you in chapter 3 and 4, and then we'll go over all this in class. If you have any questions, the sooner you send them to me, the better, because the exam will be coming up shortly.